Thank you, Brian, for the kind introduction. Again, my name is Derek Urwin. I'm a captain with Los Angeles County Fire Department, proud member of Local 1014, also an adjunct professor in the Department of Chemistry and Biochemistry at UCLA and in the Cancer Control and Survivorship Program at the UCLA Johnson Comprehensive Cancer Center. Also serve as Chief Science Advisor at the International Association of Firefighters. That probably all sounds like a contradiction in terms to you, but there's a backstory there. I lost my younger brother Isaac to leukemia when he was just 33 years old. And it was my fire service family that got me and my family through those tough times. And after things quieted down a bit, I decided to go back to school to study chemistry to understand how chemical exposures can drive cancer risk. And what I'm going to talk to you a little bit about today is research that we have ongoing here in the state of California with the support of the IAFF as well. That is designed to reduce our cancer risk. But before I do that, I just wanted to mention, you heard me mention the UCLA Johnson Comprehensive Cancer Center. You're probably aware, obviously, about our University of California campuses, the associated medical centers, but what a lot of people aren't aware of are the NCI-designated comprehensive cancer centers that we have here within the state of California. There's only about 50 of them throughout the United States, and we're fortunate enough to have 5 just in the University of California system. And the purpose of the NCI-designated comprehensive cancer centers is to conduct multidisciplinary research focused on developing new and improved approaches for preventing, diagnosing, and treating cancer. And I really want to emphasize that point, right? Research has to be done not just to understand why we have an increased incidence of cancer, but to be more successful when it comes to treatment outcomes. So after joining the Comprehensive Cancer Center at UCLA a few years ago, we were able to establish the California Firefighter Cancer Research Study, utilizing funding from the University of California Office of the President, CAL FIRE, and also the IAFF, with the goal of quantifying the biological effects of our occupational exposures and developing interventions to reduce cancer risk. And in particular, in the aftermath of the January fires at the Eaton and Palisades, we have a particular focus on understanding those exposures because they're clearly substantial. So to dive into it, I'll take a minute and start talking about what's going on underneath the hood when it comes to cancer in the fire service. So as Kristen mentioned, the International Agency for Research on Cancer classifies firefighting as a Group 1 known carcinogen. That's in the same category as things like benzene and ionizing radiation that we know cause cancer in human beings. So to clarify, just being a firefighter actually increases our risk for cancer. Now, the reason for that is because of our daily exposure to multiple carcinogenic agents in the line of duty that are not experienced by members of the general public. This comes in the form of products of combustion when we fight fires. There's also PFAS in our PPE and flame retardants, as well as other factors that we deal with like sleep disruption, UV radiation, and asbestos, and the list really does go on from there. Now, we know that firefighting causes cancer, but what's not yet well understood is the biological mechanisms that are at play, right? Like what's going on underneath the hood. And that's what I'll take a minute to talk about now, right? So when we have fires that we fight, there's a number of chemical carcinogens that are on the fire ground. We're exposed to those chemical carcinogens by inhalation, ingestion, and transdermal absorption, but there's a key point there. When it comes to inhaling carcinogens, ingesting or absorbing them, they don't just affect our lungs, our digestive tract, or our skin. Those carcinogens can be absorbed into our bodies, travel throughout the bloodstream, and impact organ systems throughout the body, and can increase risk for things like brain cancer and colon cancer. Now, the process of carcinogenesis is actually very complex and, again, not well understood, and that's the reason that we do research into what causes cancer in firefighters. So our occupational exposures, in a nutshell, alter how our cells function and then increase cancer risk. So what you're looking at on the left there is basically a healthy cell, an otherwise healthy cell. And what you see surrounding it is a number of the different types of chemicals that we encounter every time we fight fire or every time we respond to a call and are exposed to diesel exhaust. Those exposures then cause effects down at the cellular level. And what you're seeing there in the middle are things like genotoxicity, oxidative stress, immune suppression, epigenetic alterations, things that basically make our cells go haywire, which can then lead to the quintessential feature of what a cancer is, which is uncontrolled cell growth and proliferation. So when it comes down to it, there's 10 mechanisms that we refer to called the key characteristics of carcinogens that can drive cancer risk. Those include the few that I just mentioned, but there's also other things like DNA damage, things that can cause immortalization of cells. A lot of people don't realize that cells are supposed to die off in your body every single day and then be replaced by new and healthy cells. And one of the things that happens with cancers is damaged cells don't die off when they're supposed to. And these mechanisms haven't been well studied in the general public, let alone in firefighters as well. So this is where the California Firefighter Cancer Research Study comes around and is so important to us as California firefighters, but also for firefighters across the United States and around the world, where we're looking at conducting research to quantify what causes cancer so that we can actually do something about it. And what we're doing in this study is examining first persistent biomarkers of exposure. So when we talk about biomarkers of exposure, we're talking about chemicals that have gotten into our body that can then increase cancer risk. And some of those chemicals are persistent, right? After you're exposed to them, they stay in your body and they wreak havoc, sometimes for years to come. So that includes things like PFAS, heavy metals. And what we're also doing that hasn't been done previously is what we call trace element analysis, where we use advanced mass spectrometry methods to look for exotic things like uranium or strontium that we may be exposed to on some of these substantial fires that we fought recently, like the Eaton and the Palisades. Now, where the rubber really meets the road when it comes to preventing cancer and being more successful at treating cancers is understanding markers of effect. And so I'm going to take a second and pause here, right? I'm going to talk about a little bit of biochemistry. When I went back to school when I was 35 years old, I was married, 2 kids, mortgage, full-time firefighter working on Truck 3 in East Los Angeles. This is something that any one of us can do and understand. We just take our fire service work ethic and we apply it to a problem that needs to be solved. Sometimes it can be tricky, but it's no different than when you have thrown every ground ladder on the truck and then you've got to throw the 35, right? You just do it one rung at a time and eventually you accomplish your objective. So when it comes to understanding things like biochemistry that's applicable to our cancer risk, as hard as it might seem, it's something that any one of us can do. We just have to apply our firefighter work ethic. And the reason we have to do that is because it's important to our health and it's important to our families so that we can stay healthy and come home to our families and enjoy a good long life. So when we talk about biomarkers of effect, we're talking about measuring the things that are actually going on in our cells that may be driving cancer risk. So what we're doing in this study is actually looking at things like DNA methylation, which can alter gene expression. We're looking at mutational frequency and mutational signatures in DNA, which can actually alter the way proteins function. We're also looking at something called RNA transcriptomics. That's something that basically governs or allows us to measure how genes are being expressed. And then ultimately, we're also looking at what are called proteomics, where we can actually measure markers of immune suppression and inflammation. Let me back up one second here. There we go. Now, what we're starting off with is what we call a cross-sectional comparison of those biomarkers of effect. So in other words, what we do is we look at these biomarkers in firefighters who, for example, responded to the Eaton and Palisades and firefighters who did not. And that lets us kind of pick out needles in the haystack that may be indicative of the biological effects of those substantial exposures that we've dealt with recently. In addition to that, what we're doing is longitudinal research, where we collect blood samples at 2 points in time. So there's basically baseline, which is before, where we, again, look at things like DNA methylation, RNA transcriptomics, and proteomics, and then we let a year go by and do a follow-up blood draw to see what, if anything, has changed. And if you look at the left side of the screen versus the right side of the screen there, let's say if everything is otherwise normal on the left, and then on the right, what we have is the effect of, let's say, 1 year on the job, of fighting fires and how that affects our bodies insofar as changes to DNA, RNA, and proteomics. One key point, anytime we talk about looking at alterations in DNA methylation or mutational frequencies and signatures in DNA, when it comes to research, we're not talking about doing like a 23andMe type of thing, right, where we're talking about like hereditary things or anything like that. What we're actually studying is the damage that's caused by our occupational exposures. That's often a concern that people have, but to be clear, what we're doing is examining how our occupational exposures damage our DNA that can then lead to cancer risk. So to kind of tie that all together, right, DNA is the blueprint for how our cells are supposed to function. So again, up there in that top left corner of the screen is an otherwise healthy cell. And then if you kind of zoom in and see what's going on, your DNA gets turned into RNA by separating out the 2 strands. And then from there, that RNA is used to create proteins. And that's basically what makes the machinery of your cells go round. But what happens is when we're exposed to chemical carcinogens in the line of duty, when we're exposed to other types of carcinogenic agents, things go wrong, right? Our DNA sustains damage, which is those red marks that you're seeing on the DNA there that were not there previously. This can alter gene expression, right? So basically the RNA that's needed for your cells to function properly is not produced. And then from there, that can have impacts on the proteins that are needed for your cells to function. And what we're doing with this research is what's called an integrated analysis of our occupational exposures and effects, right? So we're not just measuring like one biomarker and saying, oh, firefighters are all out of whack on this one biomarker. We're saying, what are we exposed to? How does that damage our DNA? How does that alter gene expression? And then how does that alter the proteins that are then functioning in our cells? And then how does that drive cancer risk? And anywhere along that way, our goal is to develop interventions to then reduce cancer risk. Now, when it comes to doing research, right, traditionally, academia has just taken research, gotten a bunch of data, published papers, presented at conferences, and kind of left the research participants with no clue as to what's going on. So one of the key components of this research project is report back of research findings to study participants. And we do that through what we call a community-based participatory research approach. So to just use a rote definition, CBPR is a collaborative approach to research where scientific researchers and firefighters collaborate as equal partners on all aspects of a research project. And there's a key point there, right? Equal partners. It's not researchers telling firefighters, hey, we're the experts and we know what to do. It's researchers going to firefighters and saying, what are the problems that we need to address? What are the questions that we need to answer? And let's work together to do that. In this study, the way we accomplish that is utilizing a firefighter community advisory board, where basically we work together as researchers and firefighters to develop research aims, to develop the study design and ultimately execute those studies, develop interventions that are compatible with our public safety duties. And that's a key point, right? Compatible with public safety duties. You know, if most researchers just say, well, why do you do that? Just don't be exposed to that. Well, we know that we're duty bound to do our job, and that means that exposures are going to come with the territory. But what the idea is, it's to develop interventions where we can perform our duty, but do what we can to take care of ourselves again, to keep ourselves healthy. And then key point, again, is dissemination of research findings, which doesn't just mean publishing papers and say, hey, go look up that paper. It's reporting research back to firefighters in methods that are comprehensible and actionable, right? And this isn't something that's just like a demand of Derek Urwin or something like that. This is something that's extremely important to the biomedical research community, not just with firefighters, but the entire public, to the extent that the National Academies for Science, Engineering, and Mathematics actually issued a multi-hundred-page report on returning individual research results back to study participants, where they coined the term helicopter research, where researchers come in, sort of take what they need, and then take off like in a helicopter, never to be heard from again. And that's the type of thing that we actually prohibit in this type of research. We will not ever do any type of helicopter research. And to that end, with the research that we started in January after the Eaton and Palisades fires, we've actually already reported back data to over 1,000 firefighter participants. And we do that utilizing something called the Digital Exposure Report Back interface, which is basically like a web portal that you can log into to look at your research results. And we work really hard to make sure that those research results are comprehensible. So what you're looking at is an example of a PFAS, a serum PFAS data report back, where we put things in context, right? We say like, okay, how much PFAS did we measure in your blood? How does that compare to the rest of the general public? How does that compare to the other firefighters participating in the study? And not only that, what do you do from the clinical health perspective insofar as additional screenings that you might need to do? And we have study physicians that can engage with firefighters whose PFAS are above a given threshold so that they can undergo additional screenings. And one thing that we're starting to look into now is the development of intervention studies that may be able to decrease serum PFAS concentrations based off of research that's coming out of Denmark and Sweden recently. So just to kind of give you a little bit of like a nutshell of what we've been able to find just in a few months of doing this research with the firefighters that are participating is that serum PFAS levels in firefighters increase with years of service. And in addition to that, higher levels of serum PFAS concentration are correlated with a greater probability of being clinically diagnosed with high cholesterol. We know from research done in the general public that elevated serum PFAS levels can contribute to hyperlipidemia, and we're actually seeing that among firefighters. Now, again, key point here, it's not just to highlight that, hey, there's this problem. We're working on figuring out what to do about it because the goal is to keep our brothers and sisters on the job healthy. In addition to that, we'll also be reporting things back in what we refer to as aggregate form. So when we look at mutational signatures, for example, in DNA, you're literally looking at like terabytes and terabytes worth of data, and we don't just dump a terabyte of data back onto you and say like, here, go figure it out. So what we do is we actually break this down where we try to understand what the signatures are for the chemicals that we're exposed to, and then we'll be able to report that back to firefighters. And we just recently finished doing what we refer to as whole genome bisulfite sequencing of about 1,200 samples. That's really unprecedented when it comes to firefighter cancer research, or really just cancer research in humans in general, where we're going to be able to actually take a look at what we call single base resolution. And I know I'm diving into some details here, but it's really exciting from the scientific perspective because it's data that we'll be able to use to understand what drives cancer risk and how to intervene and reduce that cancer risk. And that's the ultimate goal of any of this research, is data-driven interventions, right? It's not for scientific accolades, it's not for notoriety, it's not for conferences, it's not for papers. It's about figuring out what we can do as firefighters to reduce our cancer risk. And that requires data-driven interventions. And so what we do is take data that we gather from this research and we apply it systematically using approaches like the hierarchy of controls, where we figure out what exposures could we eliminate, what exposures could we substitute, how could we isolate people from the exposures that we encounter. What can we do where we change the way we work, still performing our duty, but what can we change about the way we work to reduce exposures? And then finally, there's PPE. I always like to mention or highlight the fact that these tiers in the hierarchy of controls are listed in order of effectiveness, and you'll see that PPE is down there at the bottom. So key point, proper use of PPE at all times when we fight fire, especially SCBA, is a must, right? Nobody's saying that PPE is not important. But key point is these other measures that we can apply can be much, much more effective than just relying on our PPE. And that's the purpose of doing this research. And as Kristen mentioned, we talk about drops in a bucket sometimes when it comes to cancer risk. If you haven't heard that analogy before, what we refer to is imagining taking a bucket, putting it underneath a faucet that leaks just one drop at a time, and you just kind of stand there and watch it, and you get the impression that the bucket's never going to fill because it's just filling one drop at a time. But if you walk away and stop paying attention to it and come back about 12 hours later, you'll find that that bucket actually filled and in fact overflowed. And the reason for that is because it was filling continuously, even though it was filling slowly. And so the goal of these interventions is to keep as many drops out of the bucket as possible when it comes to our cancer risk. So in summary, the California Firefighter Cancer Research Study currently has over 1,500 firefighters enrolled. Actually, that's an old figure now based off of recent enrollments. We've enrolled another several hundred the last couple weeks, where all California fire departments are welcome to participate, with a particular focus, we hope, on recruiting firefighters who responded to the Eaton and Palisades fires so that we can quantify the effects of those exposures and do whatever we can to intervene when it comes to preventing adverse health outcomes. As President Rice mentioned, we'll have our research team from UCLA here today, starting at about noon. They'll be located right next to the CPF booth, and then we'll have our research team here available to draw blood if you're interested, and also we'll be able to talk to you if you're interested in taking this study back to your department or to your members. One of the key features of this study is that we're examining multiple key characteristics of carcinogens. There's a lot of great research going on around the country and around the world looking at firefighter cancer research, looking at firefighter cancer, but there's very few that have had the opportunity to actually look at multiple key characteristics simultaneously to see how they're integrated with our occupational exposures to really understand what's going on underneath the hood. And that's a key point, is that we're utilizing approaches not previously employed in firefighter cancer research. And we're very fortunate to be able to do that with funding from CAL FIRE, with funding from the Office of the President, UC Office of the President, and the IFF, where we're looking at 43 blood serum PFAS to measure our occupational exposures or other sources of exposure to PFAS. Again, looking at DNA mutational frequency and signatures, which is something that, again, can have a massive impact on cancer risk, which really has not been studied in firefighters. You've probably heard of DNA methylation in previous firefighter cancer research. We're actually utilizing an approach called whole genome bisulfite sequencing, which allows us to zoom into like all roughly 30 million potential DNA methylation sites in the genome, which has never been done before. Previous approaches have looked at 800,000 or so. And so it's a really amazing opportunity for us to understand what is driving cancer risk. Again, RNA transcriptomics and O-link proteomics, I know that's getting in a lot of the nitty-gritty of the biochemistry, but again, as difficult as these things seem like they are to understand, it's inherent upon us to take the time, put in the effort to understand what's going on underneath the hood when it comes to our cancer risk. And again, I did this stuff when I was a firefighter in East Los Angeles, still working full-time with 2 kids, and it's just a matter of applying our fire service work ethic to deal with these types of problems. Lastly, you heard President Rice mention AB 700, and I just wanted to mention that here. We've been extremely fortunate here in California to have support from the state legislature when it comes to preventing firefighter cancer. And the California Firefighter Cancer Prevention and Research Program was established by legislation in 2023 and also followed on with $7 million in research funding to go to University of California schools to fight cancer in the fire service. And as a result of that, there's actually, I think, about 8 projects now that have been funded where fire departments are working as co-equal partners with researchers at UCLA, Davis, UC Irvine, Riverside, Lawrence Berkeley Lab. And our hope is that this is just the first round of firefighter cancer research funded by the state. Our goal is to have these studies go on for years and years to come, because this isn't a problem that we'll solve with just a couple years of research. It's a problem that we all need to be working on for the duration of our careers and the duration of the careers for the next generation to come. So when it all comes down to it, I always like to come back to this picture anytime I'm talking about research. This picture can be duplicated at every single firehouse here in California, every single firehouse across the United States, right? It's all about our fire family. It's about the people we work with. It's about the people we go home to every day. And that's the goal of this research, is to take care of that family. So with that, this is my contact information. Please reach out to me at any time. And I really do mean that. Give me a call, and happy to talk research with you anytime. And with that, I thank you for your time.